This afternoon, the first session this afternoon, my name's Tony Cunningham, by the way. You can see that on the my credits on the screen there. So this first session, we're going to talk about what goes wrong, given all the things that we heard essentially go right or interact this morning. And in particular, we're going to focus on one of our greatest challenges, and that is dementia. 400,000 people in Australia diagnosed, currently expected to double by 2060, and particularly with the wave of baby boomers coming through. And so we have Professor Glenda Halliday, who's going to lead off talking about that, then Professor Sharon Naismith, and then of course, we're going to talk about a very topical field, and that is of long COVID, a very mysterious illness with over 180 symptoms, still being sorted out in terms of the physical versus the psychologic. A lot of people in groups in Australia now working on the effect of the virus on the brain, so it'll be very interesting to have Associate Professor Lycette Cysique talk about that, particularly as the government has set up a $50 million fund for research into long COVID, and I'm apparently going to be involved in that, and being an infectious disease physician, I guess it's fairly obvious that we should have something where we know the exact start date on a subacute illness of the brain, a bit like HIV, which Lycette has worked on before. So the first speaker is Professor Glenda Halliday, who's a legend in the field and is a fellow of both the Academies of Science and Health and Medical Science. She's an NHMRC Leadership Fellow and has been an NHMRC Fellow since 1990. Was in the School of Faculty of Medicine and Health and School of Medical Sciences, like myself, at the University of Sydney, and her particular interest looking at the structure and function of brain in illnesses that cause dementia, and also looking at how they impact on the diagnosis of such neurodegenerative diseases. And Glenda has won many awards. I'm not going to read them all out. They're in the program. She'll be relieved at that. So without ado, Glenda, would you like to address us?
Thanks. And I'd first like to acknowledge the Gadigal people of the Eora Nation on whose lands we meet today. And I pay my respects to their elders past and present, and to all those Aboriginal and Torres Strait Islanders that are here today. I thank you for giving me this talk. I must admit, for 10 minutes, I really can't tell you about anything, okay? So my specialty is in neurodegenerative diseases, and I'm especially not telling you anything about that except in a very broad sense. And the reason for that is that you can ask me any questions in the question time. So what I am going to concentrate on telling you about is actually how your brain ages and the cellular consequences of the ageing process on the brain. So there's a few myths about brain ageing. Some of them are that there is substantial brain loss, and that's a normal part of ageing, and that all protein deposition in the brain, I mean, George has actually suggested a couple of times today, is abnormal. And because of these changes, we can all expect to have significant intellectual decline as we age. So I'd like to, at the end of this, show you that these are myths at the moment. We actually don't have good evidence for any of them. So just, you heard about body size in terms of animals and how big a brain can be compared to the animal size. And it happens in humans that your brain is actually related to the size of your body. And it looks like as we age that there's a reduction in the brain size, but these are cohort studies. And in cohorts, as we actually, I don't know if you've noticed, the children are actually often taller than the adults. And it depends on the environment. If you've gone through deprivation during wars and things, then your body size will actually have an impact. It'll have an impact. So your brain size is directly related to body size, but it doesn't necessarily mean that older people have reduced brain size compared to what they were 10, 25 years ago. So what determines brain size? In a human, is it actually bigger cells in the brain? And therefore, with age, people who have bigger cells actually might have more trouble with the metabolism of a bigger cell? Or is it that there's more cells? And it's actually more cells, and this is the first set of studies that we did quite a long time ago, where we showed that the size of the brain related to the number of cells in the brain. And since then, there's actually quite a number of studies that have actually shown the same thing in humans. And some of the myth that we had beforehand, I don't know if you can see on the years of reports on the left-hand graph, which thinks that we have a reduction with age, it's because of the methodology that we used in those very early studies. And with better methodology, after a certain time point, you can see that the studies show there's not a loss of brain cells with age. So the loss of neurons, when we measure those things, is more likely to be due to a disease trigger rather than just ageing itself or abnormal protein deposition. So even though I said that you don't lose neurons, you heard from one of the speakers this morning that there is a change in MRI and brain structure, and there is. So you might not lose actual numbers of neurons in the brain, but the connectivity, the myelin, the white matter, actually is the place that reduces with age. So you actually do have a reduction of the structure of the brain with age. And so this suggests that the main thing that might happen with age is the connectivity of the brain actually changes. Now, there are actually many cells that are actually necessary for brain connectivity, and in the white matter, neurons are not one of them. Neurons have axons that go through the white matter, and the white matter actually is mainly made up of glial cells. And the glial cells do a number of things. There's microglia, which actually get rid of debris in the brain, and there's oligodendrocytes, which insulate the axons so that they can conduct the electricity faster to get to the synapses to have the messages go through. So, and then there's astrocytes that actually tile the entire brain that are necessary to deliver the oxygen, the glucose and everything that every cell needs in the brain. So there's a number of brain cells that are actually not neurons that are really important for maintenance of the white matter. And it looks like these are the cells that are actually going to be most affected during ageing and actually are going to affect connectivity. And there's some studies now, I'm going to go through some of the studies that are now coming out using quite new technologies, this is genetic transcriptomics, showing mouse brains over time, where we have done most of our research on the brain is actually in the mouse brain. And these studies, this is a recent study, only published in 2023, and it shows that the glia, rather than neurons, have a widespread ageing signature, and that the neurons themselves didn't actually change over time with age in the mouse brain. And the molecular ageing was particularly accelerated in the white matter, which is where in humans we measure age-related changes most. There's a 10-fold increase in age-related signatures in the white matter compared to other brain regions, particularly cortical regions, which we've been talking about. In the white matter, the microglia are most affected, but they're not the only ones. Mature oligodendrocytes, which we discussed, the endothelial cells, astrocytes, as well as progenitor cells are all affected. And there's particularly a mitochondrial downregulation, and the mitochondria are the energy of every cell. So within the white matter, they're the things that are mainly affected. Now, these glia in humans are actually much larger. Their microglia are relatively conserved, but the astrocytes are actually much more larger than in mouse and much more complex. And on the pictures on the right, the smaller pictures are actually rat astrocytes, and those really complex big pictures are different chemical reactions seen in astrocytes to show their structure. And you can see that they're 15 times larger in volume in a human. They have 10 times more processes. There's actually subtypes of astrocytes in humans and primates that we don't see in other animals. And they enhance higher level neuronal functionality. So they're really, really important, these cells. The compact myelin is substantially increased with the increase in processes of the astrocytes. So there's a direct connection to the connectivity, the electrical conductance through to neurons. So they're important cells. So glia are enlarged, are actually more complicated in humans compared to the animals, and therefore you might expect that there might be even a greater age-related effect on these more complex neurons, cells. So the white matter glia do quite a lot of things. There's a cartoon there that shows some of them. You can manipulate neuronal activity, both chronically and acutely, using bioelectric networks, and the bioelectric networks in the brain are actually maintained by these syncytium of glia. The astrocyte networks can increase activity. They couple the activity of populations of non-synaptically connected neurons via chemical messages, ATP secretion and calcium elevation. And they affect the dependent myelination that maintains our connectivity. And the microglia on the reverse side can suppress neuronal activity through some of the same mechanisms, and they limit activity-dependent myelin growth. So there's these yin and yang of how these cells might be affected, and they actually connect. We think of neurons as having connectivities that are direct, and you heard Lucy this morning with a lovely talk about looking at those things. These cells actually can manipulate multiple non-connected neuronal networks in a different way. So that's what happens when we age, and there's a group of cells that people don't usually talk about that in humans are actually quite substantial. We have a lot more myelin than nearly anything else, and you might be interested to know that during the puberty years, that's when our myelin is most developing. So some of the other things that we've been talking about today, myelination may have a direct effect on. These glial cells may be having a direct effect on. But I'm going to talk about how this might relate to the loss of the neurons, which is actually related to diseases of the brain, the age-related degenerative diseases. So there are age-related proteins that deposit in the brain, and George was alluding to some of these. The two most common are the A-beta peptide and the tau protein. And the A-beta peptide isn't actually found in neurons. It's not found in any cells. It's found extracellularly. And so it actually accumulates as in the extracellular spaces rather than affecting any particular neuron, any particular cell. You might be dismayed to know that at least 50% of the population surviving to older age will have an extracellular deposit of these A-beta peptides. But not 50% of the population has a disease of the brain. So, and I'll show you some data in a little while. Tau's the same. It's a microtubule-associated protein that's important for your axon stability. And most people, even young people, will have some tau deposition in their brain. So I'll show you some of the data. So the protein depositions do occur in the brain with ageing, but not all of the people that have protein depositions will have any significant neuronal loss. Neuronal loss is a prerequisite for a neurodegenerative disease. And so this is two maps. On the left, I've got a map of the normal age-related tau distribution, and if you have a look at 50% of the population will have some tau deposition at the age of 50. So I assume everyone in this room, some tau deposition. And I don't think it's bad, it's how much you have that's a problem, not how little. So, and by 70, 80% of the population will have some tau deposition. So this is really a normal age-related protein that deposits in some of the neurons in our brain, and we can all look forward to having it, and it starts as early as before 20. So people can have it very early. It doesn't necessarily mean they're going to get a degenerative disease. It's how many you have. You have to have lots of tangles to have a dementia. And we can say exactly the same about the extracellular protein, A-beta. At 50% of the population, by the age of 70, will have some of that in your cortex. So again, it's not something to actually, it's an age-related protein deposition. It's how much you have and how it might affect the neuronal populations associated with dementia. So a little bit of the abnormal protein is probably not enough, and it's the amount of protein, but also other things that happen. So these are two detailed studies, which I'm not going to go into real detail about, that looked at people who had only mild cognitive impairment. And some of them didn't have any cognitive impairment, but they followed them over time, and these people are thought in the literature to have a lot of these protein depositions in the brain. But 74% did not have enough protein deposition in the brain to think that they would have had a neuronal problem like Alzheimer's disease. More had cerebrovascular pathologies, and they did have tau, but as I said, lots of people have tau. The 22% had other pathologies and 25% only had some Alzheimer-type pathologies, that tau and A-beta. So the majority actually didn't have what we would have expected to have as their primary underlying pathology associated with what people think of as dementia in terms of Alzheimer's disease. This suggests, and it's much more prevalent, that just having the tau and the A-beta is not sufficient, and that in most instances you will have multiple proteins going wrong or having a vascular problem as well. So you have to have multiple things happening in the brain for it to have an effect on a degeneration, neuronal degeneration. So it's the severity and the numbers of pathologies. Now the other thing is that, that there are distinct glial changes that also happen in these neurodegenerative diseases, which no one really talks about that much. And in fact, the glial pathologies are more likely to be distinct between the different types of diseases. So, for example, on the top left, those are inclusions of alpha-synuclein protein in oligodendroglia, which alpha-synuclein is a synaptic protein, and these are the myelinating cells in the brain. So we have a protein in an abnormal position in cells that we really usually wouldn't see these things. On the right there, they're astrocytes in patients that have Parkinson's disease. Now the neurons in those areas, you can see a nice neuron in the bottom, big cell, with the astrocytes with the abnormal protein. So the astrocytes in this area are probably not working properly because they have an abnormal protein. The neuron is probably not working properly because its astrocytes are a problem more so than the neuron itself. So glia are affected, and then there's actually completely different types of tau that happen in different types of the astrocytes in the brain, and these are all different types. The top are called primary tauopathies, so these are astrocytes The actual disease, neurodegenerative disease, is defined by their astrocytic pathology. At the bottom, it's called RTAG. But RTAG is a very familiar pathology now that's seen in chronic traumatic encephalopathies. So you'll hear a lot more about astrocyte changes that are to do with longer-term cognitive changes in the brain of people that have, I would say, environmental impacts in terms of head injuries or sporting injuries. So, glial cells are the most affected in the brain. You probably haven't heard of glial cells in this symposium yet, but they're the most affected in the brain with ageing. But neuronal loss and a lot of the cognitive changes are actually because neurons don't connect, don't fire properly, and the neuronal loss defines degenerative diseases. So if we now think about what this might mean, brain ageing mostly affects the white matter, while neurodegeneration mainly affects grey matter structures. And in degenerative diseases, it's regional, so the type of neuron or the function of the neuron is actually important. But mechanistically, that suggests that the connectivity between regions is probably really key to disease vulnerability. So ageing, these are all age-related diseases, these neurodegenerative diseases, so having a problem with the white matter and the glial connectivity is a predisposing factor. Humans have evolved larger syncytiums, particularly of astrocytes and myelinating oligodendroglia, and mechanistically that suggests that these syncytiums are actually really important for cognitively and something that really hasn't been looked at so much. Age-related neurodegenerative diseases all have abnormal protein depositions, and the neurons are largely resistant to increase amyloid and tau with ageing, and mechanistically that suggests that there's additional cell or tissue changes that need to be involved to have neurodegeneration. There are distinct glial cell pathologies, and these define most neurodegenerative disease, which is largely still ignored. Mechanistically, this supports important roles for glia in neurodegeneration, suggests that pathological dysfunction of different glia impact differently on these neurodegenerative diseases. And I'm looking forward to the other talks and happy to take questions later.
So the next speaker is Professor Sharon Naismith, who's the Leonard P. Ullman Chair in Psychology at the University of Sydney and also an NHMRC Leadership Fellow. And she's really going to expand on what Glenda has laid the foundation for, because she's a clinical psychologist and she's in particular interested in risk factors for neurodegenerative diseases of various types, and I'll let Sharon tell you more about that. Thanks, Sharon.
Thanks very much, Tony. I'm very honoured to be here today amongst such distinguished guests, and also very honoured to be on this beautiful Gadigal land. So thank you, Glenda, for setting the scene on dementia. So I will fan out from here and really just wanted to bring to your attention some key issues and opportunities we have in the field at the moment. It's a really exciting time to be working in this field, but we do have many challenges ahead of us in terms of how to deal with the increasing number of dementia cases that are set for us. So globally, there's about 50 million people with dementia. There's 400 to 450,000 in Australia. But there's an extra 1 million people that have what we call mild cognitive impairment. So this is what could be deemed to be a prodromal stage for dementia. So if you have mild cognitive impairment, you show cognitive deficits on the awful tests that us neuropsychologists give you, and about 45% will progress to dementia within 5 years. And this is really an important period because this is actually an opportunity for us to think about prevention strategies. Dementia now actually overtakes heart disease as the leading cause of disease burden in those over 65. And interestingly, a third of the dementia population live in regional and rural areas of Australia. And the prevalence of dementia is actually 3 to 5 times higher in Indigenous Australians, and they also get dementia earlier. And $3 billion of Australians' health and aged care expenditure is spent directly on dementia, and there are lots of other indirect costs as well associated with it. So I just firstly wanted to talk about prevention, and Glenda told you about the different types of dementia pathologies. So this is the umbrella term that we use for all the different types of dementia, but actually there's a couple in particular that I'll focus on in this talk, and that is Alzheimer's disease and vascular dementia, because these are the ones that where we know that there are actually some risk factors that can be modified. And so there are two key things to think about when we're thinking about dementia prevention. Again, everyone in this room should be thinking about dementia prevention. We all should be thinking about dementia prevention from midlife, actually even earlier. And certainly we know that the pathological changes that occur in the brain prior to dementia are building up 10 to 20 years before someone ever attends a memory clinic or presents to their doctor with symptoms. So you can see here there are many changes. We don't actually have the glia on here, Glenda. We'll have to amend this graph. But we have many changes here in amyloid, the sticky kind of plaque substance that builds up in the brain in Alzheimer's disease. We have changes in the synapses of the brain. We have tau accumulating, and then we get changes in the structure of the function, and then eventually you'll see in this MCI stage here in the middle with the blue lines and the purple lines that you then get changes in cognition. And so what we know about this is actually about 40% of the risk for these types of dementias is due to things that are modifiable. And these can be mapped across the life course. So there was a great paper in 2020 in the Lancet commissioned by Jill Livingston that identified all of these risk factors, and that had built on prior work that was epidemiological work looking at the population attributable risk of these risk factors. So big ones we really need to consider are depression, also many different types of diseases that are linked to cardiovascular disease. So hypertension in midlife, cholesterol in midlife, also alcohol. So the epidemiological data suggests more than 21 drinks a week. That's actually a lot. And so many people are relieved by that in relation to dementia. But some of the imaging data suggests actually that perhaps it's a little bit more nuanced than this, and in particular alcohol may affect the white matter, and we should really be thinking about much lower levels of alcohol than that. So the jury's out a little bit on that. Other factors are obesity, smoking, going back to the discussions we had about social connectedness and social networking in urban planning, etc. These are all important. And new ones that have emerged recently are air pollution as well, accounts for about 2% of dementia risk, as well as hearing loss. And indeed a recent trial showed that if people have cognitive impairment and hearing loss, then the cognitive impairment could actually be slowed. So it's important to think about all of these risks if we want to take a public health perspective for dementia. Indeed, we think dementia prevention's everyone's business, so we should really be thinking about how to create guidelines for dementia prevention. We know a lot in the media about heart disease and what we should do to prevent it. In dementia, the best thing we can do is say, follow what the Heart Foundation say, because everybody knows that. But no one knows that dementia is actually a multifactorial disease, and we really need to be educating the public about what we could be doing. We need to be able to implement in a memory clinic setting what people could do to slow their disease and to build a better evidence base around that, and to involve consumers in that, people with diverse cultural backgrounds, Aboriginal and Torres Strait Islanders. Really we have a lot to do at the systems level to improve the services that we provide. So you may or may not have had experienced someone with dementia going to a memory clinic, but typically they're told to go away and get their affairs in order, and not much is provided to them after that. So we really need to improve what we're doing around health advocacy and working also closely with governments and with policy advisors generally, and have greater kind of connections with the non-government organisations like Dementia Australia to get this messaging out. But we also have some new challenges and opportunities in the diagnosis, treatment and care for people that have established dementia. So once you already have symptoms of cognitive decline, you might typically go to a GP, or if you're lucky, you'll get to go to a memory clinic and have a much more detailed assessment. We've realised that in order to tackle this on a national level, we really need to unite what we're doing across the fields of diagnosis and treatment. And for this, we're very fortunate to receive a grant by the NHMRC for $18 million to bring together many researchers working in this field across Australia. And we established a clinical quality registry, also a screening and trials program working closely with pharma, and also a memory clinic setting, which is more health system service focused. And the Clinical Quality Registry has been running now for about 3 years, and it got continued funding from the government to essentially create a register of people presenting to memory clinics with mild cognitive impairment or dementia. This was the annual report that came out a couple of years ago. We now do have 77 sites across Australia that are participating in this so that we can track the quality of care provision to people presenting with cognitive decline, but also so that we can feed that back, so we can create a loop of feedback to continuously improve services for people that are going to a memory clinic or indeed experiencing any type of diagnostic services. And so this is really to benchmark the clinical practice. It includes closely people living with dementia and also their care partners. And we have a component where we actually do get feedback about the service that they've had. And data from other initiatives internationally have shown that this actually does drive improvements in care. So data is provided back to each clinic that participates and in turn goes back to their service providers, etc., to change staffing levels, change the way things are done or change the systems. And also to measure the impact of new treatments on progression as well. And so far we've managed to look at the waiting times across the clinics in Australia in terms of how many clinics are able to offer appointments within 90 days. 90 days is actually a really long time internationally in the UK, Canada. In other parts of Europe, people get appointments within 6 weeks, and that's considered their gold standard. We got a lot of pushback from the clinicians in Australia around this because they know it's just not feasible. Our memory clinics run like half a day to a day a week in many of them, but in Europe they run every single day of the week, and I would advocate that that's the scale that we need to go to in order to reduce our waiting times and certainly increase our capacity. So there's a number of markers that we achieve and also look at the composition of who's actually attending the memory clinic. So about 30% of people now attending memory clinics are people with mild cognitive impairment, and also the cases that are seen in the memory clinics are typically more complex cases. They might have non-Alzheimer's forms of dementia or might have much more complicated history. Many people in the room here are used to running memory clinics and so very familiar with these kinds of presentations. But it's certainly expected that the bulk of dementia presentations will still occur in primary care. So the Memory Clinics initiative works closely with the Clinical Quality Registry team, and I guess the role that we have is to work closely with government to try to increase services for people attending memory clinics. So to date, we've had no national guidelines of how memory clinics should run. So we've had some very large kind of consultation, stakeholder meetings with clinicians, service providers, policymakers on how should a memory clinic run, what should be the waiting times, what should be provided. You'd be surprised to know that actually it wasn't mandated that any follow-up was provided for a person diagnosed with dementia. So now we do have guidelines around that. And also looking into how can we actually improve post-diagnostic care. So we've looked at what kind of care is provided when someone attends the memory clinic, and despite spending hours with a neuropsychologist doing assessments, actually no interventions were ever provided. And so there has been some work. Actually, we conducted over a decade ago at the Brain and Mind Centre looking at cognitive interventions and do they actually help older people that are experiencing cognitive decline. There's now a massive evidence base for these. So meta-analyses around the world show that you get about a 0.4 effect size improvement in memory with cognitive interventions, yet people in memory clinics cannot access these. So we really have a massive evidence to practice gap existing in these areas. And for the first time, we've mapped all the memory clinic services across Australia, and these are available on the ADNET website so that GPs and also people experiencing cognitive decline know where to go if there are any out-of-pocket costs, and also what kinds of services they can expect and what kind of languages services are available in. Interestingly for our field, we've actually had some advances in diagnostics. So the typical way to get a definitive gold standard diagnosis for Alzheimer's disease is actually to not only have all of these clinical assessments, but to have a PET scan looking at the amount of amyloid that you have in your brain. So as Glenda said, well, you'll have it, but there certainly needs to be a threshold that you would reach that would give you a diagnosis of Alzheimer's disease. And a clinical diagnosis would be the clinical symptoms plus evidence of the disease on PET scans. And these have been largely inaccessible. So in Melbourne, for example, you can get PET scans, but you can't get them in Sydney. There is one centre that will do them clinically, and it costs about $1,500. So it's certainly out of reach of many older Australians. But there have been advances now in looking at blood samples to be able to detect Alzheimer's disease in the blood. And so Glenda told you about tau, which is a very important protein, which is associated with transport in the microtubules. And in Alzheimer's disease, the tau is hyperphosphorylated. And it's aggregated and mislocalized. And actually you can find fragments of tau in the blood, and even though we're looking for Alzheimer's disease and the earlier signs of that are amyloid, it actually correlates very well with the amount of amyloid that you'll see on a PET scan. So we can now actually use these levels that they're quite specific for Alzheimer's disease, but we can use these levels to determine if someone does indeed have Alzheimer's disease and therefore have greater diagnostic accuracy. And this is important because there are new treatments that are coming along that do target amyloid specifically, and so it's really important that we get the diagnosis right. So typically about 40 to 60% of clinical diagnoses in memory clinics can be wrong. Because we don't really know what the pathology is underlying that. And as Glenda's already said, often cerebrovascular pathology underlines someone's dementia. It's not necessarily always Alzheimer's disease. So at the Brain and Mind Centre, we're actually leading a trial where we'll be looking at implementing these blood-based biomarkers into the memory clinic. We want to have a look at what is the impact on clinicians' diagnosis and management of the disease, and that's really important for trying to advocate for reimbursement of these kinds of tests for Medicare, etc., and to be able to roll them out across Australia and importantly into regions where they don't have good services. The other big development in the field is that we now have drugs for Alzheimer's disease. Lecanemab was actually, the results of this trial actually released last year in the CTAD conference in San Francisco. It was a very exciting time for the field because for the first time one of these anti-amyloid drugs was given and shown to clear the amyloid from the brain as observed from the PET scan, but also to slow the rate of cognitive decline by 27% and slow the changes in quality of life by 35%. So there were lots of other biologic markers of changes in the disease course. I guess the big concern with these drugs is that there are side effects, so you can get bleeding in the brain, which is a big concern for us at the moment. But nonetheless, it has now got FDA approval in the US and it is being rolled out in the US across many centres there. Also approval in Japan, and I believe one place in China has approval as well, and it has been submitted to the TGA in Australia, and in 2024-25 will undergo evaluation for PBS reimbursement. Later, donanemab was, the results of the donanemab trial was released, and this is the Trailblazer 2 studies. This had 1,700 people across 8 centres. And on the same measure that was used in the lecanemab trial, it actually slowed cognitive decline by 55%, but they used a different outcome measure. And they also showed that if you equate it to kind of a delay in clinical presentation, about 4.4 months delay in disease course was achieved by the drug, and 47% of people remained stable on the drug, whereas in the placebo group it was about 29%. Regardless of that, there has been some question about how clinically meaningful is this change in disease course. There is a risk of the bleeding in the brain. It has to be monitored very, very closely. You need to have an MRI scan at the outset and a PET scan or a lumbar puncture, which people in Australia don't really like to do. It's done very commonly in Europe and in the US. But actually you need to have infusions every fortnight and you need to have about 5 MRI scans over the course of the treatment. The treatment without PBS approval will be about $30,000 a year, so it's quite significant investment, and certainly the cost to the health services, administering and coordinating these is significant. We don't really know yet in Australia what our capabilities are to be able to deliver these drugs. We think there are probably very few centres in Australia that have the combination of PET scans, CSF scans, clinical trials, suites with infusion capabilities, expertise in neuroradiology, as well as the patients coming through as well. So I think we have some time to get used to this, but we will need to think seriously about if this does get approved by the TGA, how do we actually roll it out to Australians, and even more so if it gets listed on the PBS. A bigger problem actually is that we have massive delays in dementia in Australia, so it takes someone about 3 to 4 years from the time they first present to primary care or tell a doctor about their symptoms to get a diagnosis. Some people might say, well, why would you want to get an early diagnosis of dementia? But the data actually shows that it is important. People do want to know. It informs their choices about their future, including financial and legal matters and other things they may want to achieve in their life. And also data shows if you get an early diagnosis, and particularly if it's a multidisciplinary diagnosis such as you get in a memory clinic, you'll have a longer duration of independent functioning at home and delay admission into aged care facilities. There are many contributors to poor diagnoses, including poor awareness of symptoms, the stigma associated with dementia and reluctance to seek help, also therapeutic nihilism, difficulty recognising dementia and limited access to specialist expertise from GPs as well. About 50% of cases in primary care go undetected. As you might imagine, GPs are very time pressured. They don't have very good tools for detecting dementia. There's a lack of specialist support, as already said, and we've done a bit of a deep dive into the Medicare items for dementia, and really it's quite impossible for GPs to make any money out of diagnosing someone with dementia. In terms of what their reimbursements are, in terms of needing to talk to family members and knowing what tests to get and the time taken. So there's not really many incentives for GPs to do this. The memory clinics, of course, are better set up for this. It is expected that we will start to see an unprecedented demand, not only because we have the better ability to detect Alzheimer's disease using the blood test, but also because of the drugs that are coming along. We have actually mapped in Australia what our capabilities are in terms of memory clinics. There are only 54 publicly funded clinics across Australia, and we estimate that this probably serves only about 4.8% of the population with mild cognitive impairment, and that is health-seeking people with mild cognitive impairment. This is not the community-based prevalence studies. And if you add in the private clinics, then we still have an unmet need of about 87%. And this is not including people in the 50 to 65-year-old age range who also have the earlier onset dementias and may also have a preclinical Alzheimer's disease emerging in the brain. And certainly the drug studies, the anti-amyloid drugs, are beginning to target people earlier and earlier. So the clinical trial data shows that the earlier you give these drugs, the better the treatment response is. So it may well be the fact that we are targeting people in the future before they even get any symptoms and even come to the doctor, but they have amyloid in their brain. The other big bottleneck is, of course, by region. I said at the beginning that a third of the dementia cases are in regional areas. We only have 10% of our memory clinics in regional areas. Hardly any clinics have access to neuropsychology, so certainly no capacity to detect people with mild cognitive impairment. And certainly many of our colleagues say that it may only be about 0 to 5% of their cases as compared to the 32% that you'll see in the metropolitan areas. So as you might expect, the presentations occur very late. Often people in regional areas, the doctors there, receive referrals very late when they have behavioural symptoms of dementia, and really there's little that can be done for someone at that particular point in time. It's very reactive kind of services that they're getting. We are working with the Department of Health on developing some new virtual memory clinics so that we can actually conduct hybrid models of virtual assessments combined with face-to-face assessments, and we'll be starting our first trial of this in Echuca in Victoria and then expanding into 3 areas of New South Wales as well as South Australia as well. So the other last part of ADNET is actually the Screening and Trials Initiative, and this is really working closely with pharma to recruit people from the community that are concerned about their memory. We've now characterised about 1,600 people that have, it says 1158, but now 1,600 people that have had in-depth phenotypic assessments using MRI and PET scans, etc., and also very detailed cognitive tests. And this is because we're usually not well placed for conducting clinical trials in Australia in terms of recruiting people. So it is a database of people that then can be offered the opportunity to participate in clinical trials very quickly. We also have a volunteer portal. People can sign up to engage in research, and we're doing a lot of work in kind of trying to better understand these new plasma biomarkers and how could they be applied at the community level, and ultimately if they could be applied in primary care, that would be perfect and certainly help the roles of GPs much more. So lots of considerations for health services and policy planning. We need to think about our workforce. We need to think about dementia prevention and earlier screening. We need to think about how do we give people better diagnostic support, offer cognitive interventions and other interventions in the memory clinic setting. We need to think about greater diversity of the people that we service and how to reduce stigma in the community so that more people do come forward when they have early signs, and working very closely across primary care and allied health as well. So in summary, we have many challenges and also some great opportunities. We're at really at the kind of beginning of some exciting developments in the dementia field. It's very, very early days, but I think within a decade or so we should have developed some of these things much further and be ready to better treat people earlier and also save the government lots of money, which of course they always like to hear about. So I'd like to thank the team at ADNET and also the team at the Brain and Mind Centre and Charles Perkins Centre for their support. Thank you.
Thanks, Sharon, for a wonderful overview. I omitted to pay my respects to the traditional owners of the land, so I'll address that now. Next speaker is Associate Professor Lycette Cysique, who is a cross-disciplinary neuropsychologist at St Vincent's Applied Medical Research Centre and at the Peter Duncan Neuroscience Unit at the University of New South Wales and the Kirby Institute and School of Psychology. And she heads a research program into the neurocognitive complications of major infectious diseases, HIV and COVID. Thanks very much, Lycette.
Thank you very much for having me. I feel honored and privileged to be talking in front of this well-informed, I think, and educated audience, and also, high achievers. I mean, I've met some of you at the lunch, and some of you are scientists, others artists. So it's really a privilege for me to be here. The other thing that I wanted to do, of course, is to acknowledge the traditioner of the land, the Gadigal people, on which we meet today, present, future, and you want me to talk a bit closer here, and future leaders. I was told that I could talk in French, but I'm not going to do it. All right, so I want to talk about viruses, a little bit about long COVID and dementia. And I just want also to acknowledge before me, for me, I'm the legend in the field, Glenda and Sharon and Tony and George, and I'm really happy to be here among you today. It means a lot. I admire you a lot. Anyway, so let's talk about viruses. So I think you've done some of the job for me, Glenda and Sharon, in the sense that when we talk... How does it work? Oops. If I speak in French, isn't that going to help, is it? Oh, it's the other way around. Oh, there you go. So it's working now. Sorry, it's me. I should have practiced. So we've just talked about risk factors of dementia, and unfortunately, we have to add viruses to the list. And so they've been a little bit under-considered compared to their actual, we see them a lot. They're around, and we've learned that with COVID. And so there's good evidence from basic science that they're here and they're causing brain damage. But it's actually very hard to prove. There's competing causes, like we've just heard, but also when we look at retrospective studies of people who had a certain viral burden, it's hard to link the two. To why people would have cognitive problem now versus having a certain level of dementia burden. So I guess with COVID, unfortunately, we have a chance to look at that. So the evidence from basic science has already accumulated quite a lot, and you will recognize some of the viruses here. All of them have been linked to some level of neurodegeneration of Alzheimer's disease, Parkinson's disease, multiple sclerosis, and the specific pathway into they're actually impacting the brain. And so the area where I've worked the most are hepatitis C infection, HIV, and COVID, and or SARS-CoV-2. And those pathways are being discovered now, but there's no doubt that they also have some level of impact on brain function. So I think I really like the talk of Glenda. Let's talk about the white matter. The white matter for me is like dark energy, or rather dark matter in the universe. This is the one that we know the least, but this is actually extremely important. It's really the infrastructure of the brain, and we cannot ignore it. And so, but anyway, so talking about that, also, Glenda also talked about the fact that a lot of us do have some accumulation of amyloid beta in the brain, and that's actually a relatively normal phenomenon. And so that can be maybe in support of what we see in the field of viruses, where we see that actually amyloid beta is an antimicrobial. So its accumulation is actually in response to, it's related to an immune response. It's not there for doing nothing. That's where when we start to give dementia medication, we'll have to think about when exactly a good balance. Because in some people, you don't want to remove too much amyloid, maybe. Anyway, so that's to say here, that's the link already between viruses and neurodegeneration. So what's important, I'm trying to kind of get home, take home messages. What's important about an infection that are different from other risk factors is that they're a bit like Russian dolls. They have different steps in which they impact the brains, and they can coalesce together to make things worse. So first, infection can directly infect the brain. And if they do, and if they're not treated, they're going to be very, very severely impactful. Second, infection can actually infect the brain also through systemic infection. You don't have to have a brain infection to actually have an impact of brain damage because of an infection. You can have infection in the periphery, what we call inside the brain, and the inflammatory products actually will traffic to the brain, and they will be actually deleterious there. So, risk factor for dementia, viruses can directly cause the impact. They cause encephalitis, so a massive immune reaction with brain inflammation. They can cause things like stroke, and we've seen that with COVID. And they will also have a tendency to play with the immune system to lower the immune function and bring resurgence of viruses and pathogens that we have in the body, such as toxoplasmosis. But then, during the chronic and latent phase of the infection, things are not over. Some viruses get fully eradicated, but quite a few don't. And so during this latent phase, there is sometimes low replication of viruses, which leads to neuroinflammation. And the latency maintenance, the viability of the reservoirs, do put pressure on the immune system, including in the brain. And then this activity it can also lead to viral reactivation, like we've seen with COVID. Here, it's a busy slide, but I think the take-home message is that when we take HIV as a model of chronic inflammation or chronic infection, what we see is that over time, people are taking treatment, they're fine, but unfortunately, the virus is not eradicated, and they develop a low-grade inflammation. And we know that this low-grade inflammation is both related to what happened during acute infection and what's happening during latent infection, and all leads to higher level of cardiovascular disease. And that's very highly relevant to dementia because, as Sharon just talked earlier, cardiovascular disease is one of the number one risk factor for dementia. And what's really important in this population is that they also have low-grade inflammation because of the viruses, but they will develop on the top of that this cardiovascular risk. So they're already double whammy. But on the top of that, so I'm going to go over that one. Yeah. On the top of that, people develop premature aging. And I just had an epiphany when I followed Glenda's slide. Glenda said that aging is a change in the white matter and the glial integrity, if you want. And so that's exactly what we see in HIV. The first place where HIV actually hacked in the brain is definitely the microglia. HIV does not infect the neurons. So we see this strange level of people getting older quicker in terms of cognitive aging. So here what we see in our study is that when we compare to people of the same age, the cognitive function is lower as a function of the normal aging. And that's a little bit worrying. So you've got cardiovascular disease, low-grade viral related inflammation, and premature aging. So that's a lot of risk factor for dementia. And then, so what we see, for example, here, evidence in the brain that the cardiovascular disease does actually affect the integrity of the white matter. So it all comes together as how a viral condition can lead to dementia. I've already said something about the reservoirs. So the reservoirs are cells where the virus has integrated itself either into the DNA for HIV, but other viruses, the RNA. And it stays there, and it's supposed to be dormant. But in fact, there's the maintenance of this reservoir, and we call that a transcriptional activity. And this transcriptional activity puts pressure on the immune system, and it particularly happens as well in the brain, because there's those cells in the brain as well. And so what we've done here in our study with Bruce Brewer at St. Vincent, we've shown that the frontal white matter neuronal integrity, which exactly relates to the glial health, is actually lower as a function of the transcriptional activity of the viral reservoirs. So that's a lot of evidence for a viral infection having this low level of chronic level of damage on the brain despite being successfully treated. So I think we got to be careful there. Anyway, so that comes into COVID. So the reason why I wanted to present the knowledge about HIV is because it is relevant to long COVID. This is when I started to work on COVID, was this entire knowledge that I looked at COVID. And yet, I was a bit wrong, which is perfect in a way. So what we see in COVID, right away we saw the neurological changes, and there's a lot of them. And so when we did the study at St. Vincent's Hospital, early on, we had to be careful, people were infectious, etc., but we looked at unvaccinated people. So early on, what we see in them, a lot of anxious, depressive symptoms, a lot of fatigue. But then when we look at 12 months, we see fatigue and loss of smell and cognitive problem. So the issue with those long viral illnesses, or they fluctuant, is to tell people it's all in your head. I think it's wrong to say that to people. We have evidence that's not all in your head, especially when we look at a potential biomarker. So we looked at a potential biomarker, and the information from that biomarker came from our research in HIV. We knew there was a sort of a mediator between the immune response, the mediator, and the brain damage. And that's this pathway which is triggered when there's a high level of inflammation in an immune response in the body, and that it's supposed to be triggered, but it's supposed to go up and down quickly. In COVID, what we see is go up for 2 to 8 months. And then with this, we found that this biomarker is associated with people having cognitive difficulties. So it's early days, but that's hopefully promising, telling people actually, no, it's not in your head. You have a metabolic pathway that's actually abnormally activated. I'm going to go over that one. So that's a busy slide, but just to say that, coming back to what I said earlier, we've got acute infection that can be quite severe. So what we see with COVID, we see that when the acute activation is severe, it produces a certain kind of condition, and it's not long COVID. It's stroke, it's encephalitis, that's what I talked about earlier. When you've got mild to moderate infection, you can eventually get long COVID. Not everybody has it. And the pathogenic pathway behind long COVID are different than from acute infection, from a severe infection, sorry. But I think in COVID, we are at a stage where we need to go like, hey, wait a minute, we need to go back to the drawing board. And we need to do like we did in HIV. I mean, I wasn't there, but anyway, HIV was discovered in 1983. 1993, we had the classification. Well, we need to do the same thing with COVID. We need to go back to the board and try to find a good definition of what actually is this condition. Because at the moment, we have a lot of publication, but a lot of it is a bit noisy, messy. And so this is my luck. I just started to work with Andrew Lloyd, who should have been here in my place. But anyway, I just started to work with him at the Kirby Institute. And he actually has worked out with colleagues, etc., in the fatigue world, a research definition for long COVID. And I think that was the missing piece to my research. And so this definition basically will allow us to look at those biomarkers in a more careful way. But where the definition comes from, it comes from the field of chronic fatigue syndrome. And it's all about those mild, unexplained medical conditions that are chronic and that do produce some level of cognitive deficit, but it's mild. It's not dementia, it's mild. And they're under-recognized in the society where we live because dementia is hard to diagnose, so imagine the milder form. But anyway, so we're working on that definition. If you're interested, I'm happy to share my slide and you can go and look at the definition. Science is a collaborative work. I couldn't have done with all the St. Vincent's team. And it's also a work where you have mentors. And so that's my mentors here. They're still very important to me. They still help me today. They still help me to understand. I'm growing more every day. Anyway, I want to thank you very much for your attention.
One and a half questions. The first one is there any evidence that people had subclinical COVID that precipitated subsequent MCI and Alzheimer's? I have Alzheimer's, but I've had a loss of smell that sort of precipitated my analysis of that. I couldn't smell things anymore. I've now recovered my sense of smell. I've always wondered whether I had subclinical COVID that might have precipitated the Alzheimer's. Is that a known thing? I don't want you to diagnose my case, but I'm interested in the relationship between COVID and the smell and the neurology and the Alzheimer's smell and neurology as well.
So I think your first question is, does subclinical COVID precipitate subclinical COVID?
All right, yeah, thank you. So listen, this is not surprising to me that some viral infection could, in some people, precipitate dementia symptoms. It's related to the fact that there's already, pre-dementia, there's already some level of inflammation in the brain. And also there's potentially some level of vascular insult. So the virus will make that worse sometime. But it's not always the case. But one advice that I would give to everybody, if you're worried about that, and if you're worried about that, you should take antivirals if you have COVID. You shouldn't wait till you get too symptomatic. Okay. What I should say as well is that in resonance with Sharon's talk, all the different interventions that are relevant for different type of dementia are relevant as well for people who would develop increased cognitive difficulty because of a viral infection.
It's chicken and egg. So we all have some age-related protein deposition, which is the definition of a disease of the brain. In terms, it's just how much. So you lose your sense of smell. 30% of aged adults will have a loss of sense of smell, and so and we've known that prior to COVID and prior to other viral diseases. So a loss of sense of smell is a real age-related phenomenon, as is a loss of hearing. You lose a lot of senses. So I'm sure having COVID won't help it, any of it. But chicken and egg, I would say.
I think Bruce told me that 90% recovery of sense of smell over COVID, but often prolonged over a period of months.
Yeah, no, most people do recover. Sorry. So most people, 70% recover from long COVID at between 3 and 12 months. So what I was trying to say earlier, maybe I wasn't clear, is that at an individual level, it's always possible that someone will have a worsening of their cognitive difficulty because of a viral infection. But at the population level, at the moment, there's a lack of evidence to how often this is the case.
Thanks very much. So Glenda and Lycette, you talked about glial dysfunction and neuroinflammation. Sharon, you showed the graph of the amyloid imaging, the tau imaging, and understanding that pathway of pathology. So would you argue that we now need some kind of way to image either neuroinflammation or glial dysfunction in living people? And if so, what would that look like?
being able to measure better things in living brains would be ideal, particularly if it could give us a definitive diagnosis. I think we'd be surprised if you could measure inflammation. So everything in the brain is to do with the quantity. Like I imagine that as we have viral infections, we may have inflammation that washes in and out of the brain, and if you're good at getting rid of it, you're probably okay. And if you're not good at getting rid of it, then you'll have a problem in the long run. So in any time point, our brains are very plastic, and things will happen from every environmental impact we have. So I'm not sure if measuring inflammation, real information will give us anything. If it's there for a long time and it's of higher intensity, I'm sure it would be bad. But it'd be great to have fantastic diagnostics. I'd say rather than imaging the brain, a blood test is much better, but that would be good.
Can I just say one quick thing? So there are actually brain imaging already, inflammation for brain imaging, but there's a lot of technical issues. There's the problem of making it scalable, and also the type of inflammation that is measured. Sometimes it does not target actually what the problem is. It targets a certain type of inflammation, but not the other. So that's also the issue.
Thanks very much.